Hi guys, let's do section 3, active transport across a membrane. Active transport moves molecules against their concentration gradient, so from low to high. It is facilitated by specific carrier proteins and requires energy in the form of ATP, adenosine triphosphate. The standard example is the sodium-potassium pump, which uses ATP or energy to move sodium ions out of the cell and potassium ions into the cell, both of which are against their concentration gradients. So there's a lot of sodium outside and a lot of potassium in, but ATP is used to move even more sodium out and even more potassium in. So a shape change between in the carrier proteins facilitates the binding of two different ions. So for every 3 sodiums, 2 potassiums are moved in. Okay, so 3 sodiums bond to the protein. ATP is used, and they are forced out of the cell. Okay, and potassiums are then allowed to move in because the protein has opened up to that side. They can take their spots on the binding sites. Once they bind, it closes this end. This end will open up and release the potassiums into the cell, and the cycle continues, continually pumping sodium out and potassium in. Now, this uses about 2/3 of the total energy that your body uses in a day. So this is a major process for your cells, and if this does not work properly, it's really bad. Macromolecules are transported into or out of the cell. It requires energy. It is bulk transport. There are 2 main categories that can then be broken down. Exocytosis is whenever you're kicking something out. A vesicle fuses with the plasma membrane. The vesicle is made up of phospholipids. It'll bond with the plasma membrane and secrete its contents, just kind of kick it out. Endocytosis is when a cell engulfs substances into a pouch, which will then become a vesicle. So the cell membrane, the plasma membrane, just kind of wraps around it and brings it in, forms a vesicle, and then that vesicle is then processed. It is broken down so that the stuff that's inside of it can be used. This endocytosis can be broken down into 3 categories. Phagocytosis, which is often called cell eating. It is when large solid molecules or materials are taken in. So this would be like a white blood cell attacking something bad in a body, like a virus. Pinocytosis is known as cell drinking, and it is when a vesicle forms around a liquid or a very small particle. And then there is receptor-mediated endocytosis, which is a specific form of pinocytosis using receptor proteins and a coated pit. So basically, the cell is looking for something specific, and it's only going to take it in if it matches up with that receptor protein. Here is exocytosis. So here's our vesicle. You'll notice that it is phospholipids that are engulfing it, that are surrounding it. It will join with the plasma membrane, and once it has fused completely, these phospholipids will move out of the way and allow our molecules to move outside of the cell. Here are the 3 methods of endocytosis. So here is phagocytosis. The cell wants this molecule, wants this big old chunk of solid stuff inside, so it's going to wrap the plasma membrane around it and bring it in. Here is pinocytosis, where we have very small molecules or liquids. The membrane pinches in around it, forms a vesicle, and it moves into the cell. And here we have receptor proteins that are bonding to a very specific thing. And once they are all bonded with whatever it is that they're looking for, the membrane will pinch in and it will move that vesicle into the cell. Here is an actual diagram, or sorry, not diagram, an actual picture of it. Here we have the vesicle forming. All right. Here it's starting, so it's starting to bring the substance in, and this membrane is going to move this way. This is going to move this way, and it's going to form a circle or a sphere around it and bring it into the cell. And that's it for that section, guys. If you have any questions, feel free to let me know.